Okay, I assume you can also see the screen, the presentation. So the title of the presentation is about the CRISPR-Cas system and its use in genome editing. And at the end, I discuss some applications in gene therapy. We can start with a definition about genome editing. Genome editing is the precise modification of the nucleotide sequence of the genome. And historically, this, in order to do that, it required a double-stranded DNA break. However, I want to point out from the beginning that now the methods have improved so much that this double-stranded DNA break is not longer required, which is a great advantage. And this is due to developments in the improvements of the CRISPR-Cas system. So what is needed for the double-stranded break is a programmable nuclease, so it can make a cut in the double-stranded DNA at a certain position, and this will then trigger cellular DNA repair pathways. And there are two main pathways I can tell you from the beginning. One is non-homologous end joining, which results in random insertions or deletions. And the second one is homology directed repair, which can result in precise DNA replacements. So in order to realize genome editing, at least in the beginning, in the first methods, you need programmable nucleases that are capable to create this double-stranded DNA break at a precise position. And before the development of the CRISPR-Cas system, this was done by DNA binding proteins, like, for instance, zinc finger nucleases. But it was always very difficult to program these zinc finger nucleases to bind to certain regions to the genome. It required a lot of engineering at the level of the proteins, which was very difficult and required a lot of manpower and engineering. A little later, tail nucleases were found. For these nucleases, the rules for binding at a specific sequence were much easier, but the domains that bind certain nucleotides are relatively big. And if you want to target a sequence that has a certain size, you end up with large proteins, also with repetitive units, and so the stability of these proteins is an issue. So, and most recently, then the CRISPR-Cas system was discovered, and here the binding to specific sites in the DNA has become much easier and very easy to design, because it is based on RNA-DNA hybridization. So the only thing that is required to get a cut of double-stranded DNA in the genome is to design an appropriate oligo such that it can bind to a certain sequence in the genome. I want to also point out that genome editing, it's not an easy task. It's actually very difficult. And this is mainly due to the large size of the human genome. It's in haploid genome is 3 billion bases. To imagine how big this is, you can imagine that if it is printed in a book with a standard size font, it will result in more than 1.5 million pages, which is a size 66 times the Encyclopedia Britannica. And if you then think about that, maybe just one change in a nucleotide can result in genetic disease. So it's you can have a feeling how difficult it is accomplish the task of human genome editing. For genome editing, to accomplish, there are three things regarding the efficacy that you have to keep in mind. One is the targeting efficiency, which means the ratio of the number of edited sites to the total sites, edited and non-edited. Because if you, let's say, target a certain tissue, you can never expect that all the cells in the tissues will be edited as you want. A second issue is the correct modification. So whether you get a deletion insertion for a knockout, for instance, versus an exact replacement. And it is often so that you can get a mixture of these modifications in an experiment or in a therapeutic outcome. And a third issue are the off-target effects. So the CRISPR system may not only bind to the site that you want, but can also bind to other sites and cause non-desired effects. A few words about the origin of the CRISPR-Cas system. And this is the origin is that it is an adaptive immunity system in bacteria. On the top, you see the organization of the locus of the CRISPR-Cas system in bacteria. It consists of a CRISPR array. It's shown to the right. The CRISPR arrays consist of repeat regions, which are actually structural elements, and also spacer regions, which are actually the specific determinants of the CRISPR systems. These spacer regions will determine where the CRISPR will bind in the genome and cause the double-stranded DNA break. Another gene also encodes structural RNA. This is called a transactivating CRISPR RNA. And then there are several protein encoding genes like Cas9, which is a nuclease, which causes the double-stranded DNA break. And there are several other genes that are not really relevant for this talk, but they are involved in the maintenance of the CRISPR locus in bacteria. So when the CRISPR system is expressed, there are three genes expressed. One is the precursor of the CRISPR RNA from the CRISPR array, the transactivating CRISPR RNA, and the Cas9 nuclease. And after processing with the nuclease, you get a complex of three elements. When this complex is made, the CRISPR system can bind to the genome, to a double-stranded DNA in the cell, and then it will move along the double-stranded DNA and scan the sequence of the double-stranded DNA target until it encounters a sequence which is called the PAM, or the protospatial adjacent motif. When it binds to this PAM, it causes the opening of the duplex, of the double-stranded DNA duplex, and which gives then the opportunity for the spacer region in the CRISPR RNA to bind to one of the strands in the opened double-stranded DNA strand. And when this has accomplished, the nuclease domains of the Cas9 are activated, and the double-stranded DNA break will occur. So there are two specificity determinants. One is the protospatial adjacent motif, and then the spacer in the CRISPR RNA. So this system then was adapted for genome editing in all types of organisms, and it was simplified and to two genetic elements. One is the Cas9, or the Cas protein, the nuclease, and the second one was then a single guide RNA in which the transactivating CRISPR RNA and the CRISPR RNA were fused. So in the single guide RNA consists of two regions: a structural region, which allows it to bind to the Cas9 nuclease, the Cas9 protein, and the other one is then spacer sequence, which will hybridize to the DNA at the target. I want to illustrate how easy it is to perform CRISPR-mediated genome editing experiments. This, for instance, can be done by plasmid expression, although it's not really recommended, but the first CRISPR experiments were done in this way. So you can get this CRISPR-Cas plasmids from a plasmid repository and named AdGene. You can order it. And for instance, a typical CRISPR plasmid that expresses the CRISPR system is shown in this slide. You have an RNA polymerase expression cassette which expresses the Cas9 protein, which is used to nuclear localization signals to get the protein into the nucleus. And second, there is a RNA polymerase 3 expression cassette which expresses the single guide RNA. The single guide RNA, of course, then you need to modify. You have to clone in the target sequence for making the double-stranded RNA, the double-stranded DNA break in the genome. And this can be accomplished by restriction digestion. You can cut out a specific sequence in downstream of the RNA polymerase 3 promoter, and then insert the primer sequence that you desire, and then you are ready to go to perform the genome editing experiments in, let's for instance say, in mammalian cell lines. How is the the spacer sequence or the guide sequence selected? For instance, you have a sequence of the genome, like for instance in this slide. The first thing you do is to find a PAM motif. In this case, the PAM motif is TGG. And then you just upstream of the PAM motif, you select a sequence of 20 nucleotides, like is shown in the slide, and basically then you have the guide sequence that you have to clone in the expression cassette of the RNA that is driven by the RNA polymerase 3 promoter. There's only one thing you also have to be careful about, is that your sequence starts with a G, because this is the first nucleotide that is transcribed by the RNA polymerase 3. If it's not present in the sequence in the genome, you just add another G in the primer that you insert in the expression cassette. It even can be even more easy. There are online tools in which you can design your guide sequence or your spacer sequence. One of these online tools is called ChopChop. You can type in the target gene that you want to edit. You can select the organism. In this case, it's Danio rerio, it's a zebrafish. The CRISPR-Cas system that you want to use. Here is mentions the most used CRISPR-Cas system is the CRISPR-Cas9 system, and then also your application. Then you click find target size, and then you get a screen like is shown on the slide. You get a picture of the structure of the genome with exons and the introns and the untranslated regions, and also then a selection of guide RNAs that are you can use for your CRISPR experiments. And you also get the sequence, a list of the sequences of these guide RNAs, and these are ranked according to efficiency and to the prediction of target sites. For instance, if you see in these slides, the first sequence has, if you look at the predicted target sites, you can, there are target size either with 0 mismatches, 1 mismatch, 2 mismatches, and 3 mismatches. And in this, for this sequence, at least theoretically, there are no off-target sites with these specificities. However, this is all theoretically, of course, and even in practice, it's often observed that the CRISPR system, even if it's loaded with a specific guide sequence, it can cause off-target effects. And I will discuss this a little bit. So as I said, the wild type Cas9 proteins, they can have significant off-target activity. And I also want to point out that today there is no simple algorithm or inspection process can accurately, uncomprehensively predict the off-target substrate of a given Cas9 single guide RNA complex. Nevertheless, there are some empirical rules that you can use to improve the specificity of the CRISPR-Cas system. And a very important rule is the decrease in the activity or the lifetime of Cas9 in the cells. So if the longer the CRISPR-Cas is present in the system, in the cells, the longer there is a chance of target effect. So you have to make this time as short as possible. So delivery with an expression vector is not really recommended because the DNA will linger along for a long time. By RNA is much better because it's much less stable. And the best delivery system is then to deliver it by protein. You can buy CRISPR purified CRISPR-Cas9 protein now from a lot of companies, and you can load it then with a single guide RNA and then do your experiments with a protein loaded with the single guide RNA. There are a lot of efforts to improve the specificity of the CRISPR-Cas system, and there were efforts to look in improvements in the properties of the single guide RNA. One important discovery was that the truncating the size of the single guide RNA to less than 20 nucleotides can increase the specificity. Other efforts have focused on the engineering of the Cas9 protein, mainly to decrease the DNA binding energy. The DNA target has a negative charge, and so it's beneficial for the specificity if positive charges on the CRISPR-9 protein, on the Cas9 protein are eliminated to avoid non-specific interactions between the Cas9 protein and the negatively charged DNA. A much greater improvement still is then not to work with Cas9 system, but by two, in which that are recruited to the same locus in the DNA. One CRISPR system is recruited to the upper strand and causes a single-stranded break. The other CRISPR system is recruited to the lower strand and causes another single-stranded break in each other's vicinity. So it speaks of itself that in that case, the specificity of recruitment for of the CRISPR-Cas9 system to cause a double-stranded DNA break is greatly improved. This another strategy is to use CRISPR systems that have Cas9 enzymes that have the nuclease domain deleted, and then recruit a restriction enzyme to the targeted locus. And because it needs this restriction enzyme needs heterodimerization, it requires two Cas9 proteins to localize to the same sites, which also, of course, increases the specificity of the system. Let's go a little again a little bit more in detail to the different outcomes of the genome editing agents after it causes double-stranded DNA breaks. As one outcome is the non-homologous end joining, which causes small insertions and deletions. If there are little pieces of DNA floating in the cell nucleus, also these can be incorporated non-specifically. So this pathway is used for knockout applications of CRISPR. Another repair pathway is homology-directed repair. In that case, you provide templates which can then be replace the target. And this can be either single-stranded oligodeoxynucleotides or double-stranded DNA donors. This is a more difficult to realize repair pathway, and it requires also dividing cells. Also, the efficiency is much low. One can improve the efficiency of the homology-directed repair pathway to the expense of the non-homology end joining pathway by manipulation of the system. There has been a lot of work to inhibit the non-homologous end joining pathway by small molecule inhibitors or by gene silencing of particular proteins. And at the same time, there is a lot of research to increase homology-directed repair by small molecule activators, or by doing experiments or screenings at particular phases in the cell cycle. So the homology-directed repair pathway will is favored at the G phase in the cell cycle. So far, I have just talked about the canonical CRISPR-Cas system, which is based on double-stranded DNA break, but CRISPR-Cas has moved on, and it's now feasible to do genome editing in the absence of double-stranded DNA break. And a very popular system is base editing. In this case, the CRISPR system is targeted to the locus, and it causes the separation of the double-stranded DNA strands. It causes a nick in the strand that hybridizes to the single guide RNA, while the other strand it becomes single-stranded. And this provides the opportunity for cytidine deaminases or adenosine deaminases to modify the bases in the single-stranded DNA strand. And more specifically, cytidine deaminases convert cytosines to uracil, while adenosine deaminases convert adenine to inosine. And when the system is dissolved by later events in the repair pathways, the cytosines, cytosine guanosine base pairing is converted to thymidine adenosine for cytosines. For C base editors and for A base editors, AT base pairs are converted to GC base pairs. There are several advantages of the base editors. It's more safe, mainly because they don't cause double-stranded breaks and work with single-stranded breaks, which are less traumatic to the cell. They're much more highly efficient than the CRISPR-Cas systems. up to 35–75%. Of course, they have off-target effects which need to be taken into account. And also base editors in general, they are more limited in base changes, so because they only afford base transitions and not base transversions. Another improvement in the genome editing agents is the use of prime editing. And in this case, the CRISPR-Cas system is improved by 2 fusions. And the fusion of the Cas protein with a reverse transcriptase, and the fusions of a single guided RNA with an editing primer. I don't have time to really go into the detail, but this system is a very flexible system, and it will allow to incorporate several different small changes in the genome in one go, like one base pair in combination with a small deletion or a small insertion, and so on. So this allows more complex changes in the genome. More recently, then CRISPR has emerged as a very flexible platform for multiple applications. So I already discussed in detail the canonical system, the base editor system, and very shortly the prime editing system. But there are more other applications. Once an important application is gene regulation. The CRISPR system can be recruited to promoters, and if it's then fused with transcriptional inhibitors or transcriptional activators, it can repress or activate genes at specific loci. There are also many other CRISPR systems discovered. They can be used to cause large deletions. There are some transposases that are connected with the CRISPR system. These systems can be used to engineer large insertions at specific positions in the genome. And also the CRISPR system can be used to trigger continuous mutagenesis at certain positions in the genome. And because of the large flexibility of the system, it can also be used for genetic screens. In this case, a large library of single guide RNAs can be created, incorporated in lentiviral vectors. It can be used to transduce cell lines, and then used to assess the involvement of particular genes in physiological processes. As an example, I can give you, there are, for instance, cancer cell lines that are resistant to certain cancer reagents, and so the CRISPR genetic screens can be used then to assess the involvement of particular genes in this resistance mechanism. And to analyze it, after a selection procedure, the RNA is selected, and the single guide RNAs are it's subjected to high throughput sequencing to assess the presence of single guide RNAs. The presence of single guide RNAs can be correlated with a gene knockout. So in that case, you can identify the genes that are involved in this particular physiological process. Because of the flexibility of the CRISPR-Cas system, it also has been entered human medicine for the treatment of Mendelian disorders. It can be used for sickle cell anemia, Huntington's disease, or also certain idiopathic diseases like pulmonary fibrosis and Crohn's disease. In that case, actually, the CRISPR system is used as a certain gene therapy, and it's on its way to replace the traditional gene therapy. Traditional gene therapy was based on random insertions into the genome, which was dangerous because it could lead to insertional mutagenesis and causing cancer. The advantage of the CRISPR system that it can really correct the gene mutations in situ. Because of the nature of the system, you just need to target the CRISPR system to the locus that you want to edit. And I want to end with just an example how the CRISPR system can be used in gene therapy. The example is a mutation in the inner ear cells, in the inner ear cells that are involved in hearing. There is a mutation in the TMC1 gene in which thymidine is mutated to an adenine. And this is a dominant mutation. And it causes the loss of hearing over time in patients. So in a mouse model, they have tried to correct this and they were successful. They injected the CRISPR-Cas system into the inner ear and in the form of a lipid nanoparticle that encapsulated the Cas9 protein in complex with the guide RNAs. And what they realized was to delete this adenosine. Actually, they then inactivated this dominant gene, and which led to the improvement of the condition. Of course, not all inner ear cells were corrected. Was the mutation corrected? But this was not necessary. Only a modest fraction of the cells were edited, but this was sufficient to improve the disease condition. And with this, I want to end my presentation, and I'm happy to answer the questions.
Ευχαριστούμε πολύ, Λουκ. Υπάρχουν δύο ερωτήσεις. Η πρώτη λέει, how is it possible to minimize the off-target hits?
Yes, okay. First, you can do predictions. You select a guide RNA in which you know that are very, at least theoretically, you will have at least as few off targets as possible. And then you have to test it. Usually you can test it before you go to an animal or to the human gene therapy applications. You can test it in cell lines. So you have some candidates of guide RNAs for a particular gene, and you test them in a cell line. And there are experimental procedures to evaluate the off-target effects. For instance, a popular procedure is to use a transposon. If there are off-target effects, and they will cause double-stranded breaks, and this will provide a transposon or another element to insert in this off-target site. And so by high throughput sequencing, you can get an idea where off-target sites are located in this particular mammalian genome and at which frequency. So so it's still an empirical process to evaluate the off-target effects of the single guide RNAs. So you have to test a certain range of good candidates, and then after this experimental procedure, you proceed with the best candidate.
Ωραία. Και υπάρχει μία ακόμα ερώτηση για να πάμε μετά σε διάλειμμα. What are the possibilities and the potential of CRISPR to produce pathogens that are more lethal than SARS-CoV-2?
Okay. Well, this is a very control. I think the thing is to select. Okay, maybe you don't even need CRISPR-Cas to get a more lethal pathogen. You can grow the pathogen in cell lines and select variants that are replicate harder and cause higher pathogenicity. Of course, then CRISPR can accelerate the process. Maybe you can design, you have a library of single guide RNAs that cover the length of the pathogen, and then you select for the you select for the pathogens that survive the procedure. Anyway, it's you have to carry out a selection process to get. You need, you could, for instance, you could use a reporter assay for the pathogen, and then use as an outcome the fluorescence. Okay.